This is Dr. Launen, and from Keene State College, I'm going to talk to you about polymerase chain reaction, or PCR technology. This is a technology that was invented in the 90s and is used to specifically amplify or grow or increase a targeted and relatively short sequence of DNA. So you can't copy a whole genome this way, but you could copy most genes. It allows you to go from one molecule to one or two millions or billions of molecules. So why might you want to do that? Like, why do you want to amplify DNA sometimes? You might want to do this so that you can conduct further research or answer specific questions. For example, you can use PCR to determine if you have a particular mutant disease allele, which would tell you some information about what to expect from a healthcare perspective. You can use PCR to tissue type somebody for an organ transplant to see what genes they have and if they're a match for a potential recipient. You can look for cancer genes, which are also called oncogenes, and you can do infectious disease testing so you can determine if a sample of blood or sputum from somebody has the pathogen Mycobacterium tuberculosis and others. You can do paternity testing, forensics analysis, food or product safety, and many, many more things. In the routine research lab, what we often use PCR for is to go from just a couple of molecules of a target sequence to millions or billions so that we have enough to run a gel and then see if the molecule is there, or to send it off for sequencing, which requires a certain amount of template molecule. So this is an image of my student Katie Kiley, and she's doing PCR in my research lab, and the reason that she's doing PCR is to confirm that bacteria that we received from collaborators at the University of New Hampshire are indeed the species Vibrio vulnificus, which is the organism that we study. And she does that using a species-specific gene called the VVH gene. It's a gene only found in Vibrio vulnificus and not other bacteria. And so if she gets amplification, which she has here, you can see these a little bit blurry, but these bands of amplified product that are easy to visualize, which means there are millions or billions of molecules there. That tells us that the gene is present and that the DNA from which, the organism from which she extracted the DNA to do PCR with is indeed Vibrio vulnificus. So these are our experimental samples that are NH something, and then the VV ones are positive controls. The water should have nothing, and it does not. And the E. coli negative control actually does have a bit of a faint band in it, but we've troubleshot that, and it's also negative in most gels. And there's a ladder over on the side to show us the size of the PCR product. So we do this every day in our research lab, or many days. So to do PCR, you need to have something called a thermal gradient, which is a machine capable of running temperature extremes that change very, very quickly. You need to have gel electrophoresis equipment to analyze the products after, tubes in which to conduct the synthesis reactions, and you need to have a DNA template. You need to have DNA primers. So in PCR, we purchase specifically designed DNA primers rather than using the RNA primers that nature makes in cells. You need free nucleotides, and you need the enzyme DNA polymerase. And what's not shown here is that you also need to have an aqueous medium, which would be water, and it needs to be pH regulated and contain magnesium, and we provide that by putting buffer in the reaction mix. So the first step in PCR is called denaturing, and this is a step that is done in cells with the enzyme helicase. And instead, in PCR, we use heat. So that picture is showing two strands of DNA that are the top strand and the bottom strand, and the base pairs are being broken using heat. And so that's usually heat that's cranked up to about 95 degrees Celsius, very, very hot, and it will melt apart the template molecule that you've put in the reaction tube. The next step requires us to lower the temperature to about 55 degrees, which is a temperature optimal to allow the primers. Here's a primer, and here's a primer. This is the forward primer, and this is the reverse primer. And they are base pairing to specific regions on the template DNA. And you'll have to have two primers so that you can base pair with each strand, because your goal in PCR is to make many, many molecules. And so you're going to need to have primers that bind to each strand, the forward and the reverse strand, both serving as templates in the PCR reaction. So you need to know some information about your target gene ahead of time so that you can design these primers. Often we buy primers that other people have designed, but somebody had to design them initially, and there has to be a base pairing relationship here on the edges of the area we want to amplify. So this is the total sequence that we want to amplify, like basically from here to here, here to here. That's one gene sequence. So once you've annealed the primers, which means allowing base pairing to happen, you raise the temperature to about 72 degrees Celsius, which is favorable for the specific kind of DNA polymerase, more on that later, used for PCR reaction. So here the primers have bound and DNA polymerase is moving. This is DNA polymerase here and here, and it's adding nucleotides, so it's going this way, and this one's gone this way, and they are creating new DNA, making phosphodiester linkages between the new strands that they are synthesizing. So there's a pattern or a cycling that happens in PCR. So we initially denature the DNA by heating up to about 95 degrees, and we hold that temperature for a little while to make sure the template molecules have separated. We drop it down to about 55, anywhere from 50 to 60 really, let the primers anneal. We raise the temperature again, and we hold it so DNA polymerase can work, and that is one complete cycle of PCR. And in one cycle, we can go from one template molecule to two. So the green here is showing the new strands that have been synthesized that are complementary to the red template. And this whole time will take 2 to 3 minutes typically. So here's the same idea, and we're going to usually run PCR for 20 or 40 cycles of this. 2 thermal cycles are shown in this diagram. So we're going to run one cycle, and then we're going to repeat and run another one, and then repeat and run another one. And each time that happens, we should have a doubling of the target DNA molecules. And this allows us to have a pattern of growth that's called exponential amplification. So if you start off with 2 molecules, then in one cycle you'll have 4, and in another cycle you'll have 4 times 4, which is going to be 16, and then 16 times 16, and so on. So after 20 cycles, if you started with 2, initially you would have 2 to the 20 molecules, and you can calculate that out. But here's a visual on that exponential amplification pattern. So this is how you go from a little to a lot. And when you have a lot, you can visualize it, you can sequence it, you can study it, you can do more things with it. And this is a technique that was discovered or invented, I think invented is a better word here, by a guy called Kary Mullis. And he won the Nobel Prize in Chemistry for that back in 1993. And this is a picture of him. He's a surfer, interesting fellow. And he apparently thought of this while he was on a long motorcycle ride in the desert, although I don't know if that's kind of a scientific urban myth or not. So I want to point out that none of this could have happened, though, without earlier work done by the famous microbiologist Thomas Brock, who discovered a bacterium called Thermus aquaticus. And that bacterium grows in Yellowstone Park in the hot springs, where for a long time scientists thought that nothing grew. Let's see if I have a picture of that. There's a satellite image of, or a plane image of a hot spring. We thought these environments were sterile until in the 50s Thomas Brock thought of like dipping slides in, glass microscope slides, and leaving them there and then for weeks and then pulling them out and having a look. And sure enough, he found the slides were covered with microbial life, and he studied that life and identified the bacterium Thermus aquaticus. It's a thermophile. It likes high heat conditions, and so it has enzymes that like high heat conditions. And that's the source of the original DNA polymerase used to do PCR. You could never use human DNA polymerase because if you heated ours up to 72, it would just melt. It would degrade, it would denature, and certainly would when you heated things up to 95 degrees Celsius, which is required for PCR in order to melt apart the template strands. But the Taq polymerase, Taq is short for T and AQ, from Thermus aquaticus, puts up with this pretty well. So if you understand PCR working on your own or with a partner, you could answer these questions, and I'd like you to work on those, and we will take them up in class. And with that, I will conclude this lecture.